Okay, welcome everyone, and thank you for joining us for the ninth episode in our webinar series, From Lab to Field, Genomic, Microbial, Biogeochemical Studies. My name is Aida Morales Moreira. I'm a postdoctoral research fellow here at the University of British Columbia, working with Dr. Kara Hani. Um, First of all, as we begin, I would like to acknowledge that UBC's Vancouver campus is situated on the traditional, ancestral, and unceded territory of the Musqueam people. Each of you are joining us today from many places near and far, and I also like to acknowledge the traditional caretakers of those lands. I would encourage everyone at some point to take a moment and situate yourselves in place. On whose territories do you currently reside? How have they historically managed those lands? If you don't know whose territories you are on, we encourage you to begin that learning by doing some research to find out. One way you can do this is by going to nativeland.ca. Thank you for joining us today for the ninth installment of Farm to Globe Transforming Our Food Systems, a webinar series focusing on what has become readily apparent not only for food systems experts, but to the general public, that there is an opportunity for major benefits through some much needed changes to our food systems. We'll be exploring the most pressing issues our food systems face today and hoping to find innovative solutions to these challenges. We'll also be focusing on the social inequalities that are present and exacerbated by all of these issues. Join us for discussions that we hope will drive action and discover what steps we need to take to transform our food systems. This series is presented by Center for Sustainable Food Systems at the UBC Farm, sponsored by RBC Royal Band, and in partnership with the UBC Faculty of Land and Food Systems and the BC Food Web. The Center for Sustainable Food Systems at UBC Farm is teaching and research center as well as local to global food hub working towards a more sustainable, food secure future. Their mission is to innovate from field to fork to achieve resilient, thriving, and socially just food systems for all. The BC Food Web is a web portal project which aims to increase access and connection to current research and other resources materials. In addition, it aims to encourage innovation between farmers and researchers to enable future projects collaborations. The Faculty of Land and Food Systems is a world leader in research and in teaching about sustainable and healthy food supplies. Today's webinar topic is From Lab to Field, Genomic, Microbial, Biogeochemical Studies. We are lucky to have Dr. G.T. Cornelis and Dr. Karen Fogg here today to discuss their own work in the fields of genetics, microbiology, and soil chemistry, and what the implications of these studies are for our food systems. After each panelist gives a brief presentation, we will then move to the question and answer portion of the webinar. If you have questions for the panelists, please navigate to the question and answer tab at the bottom of your Zoom control panel and share them with us. Also, feel free to upvote others' questions by clicking on the thumbs up symbol. If you would like the opportunity to chat with our other attendees, the chat feature will be available. However, inappropriate and offensive comments will not be tolerated and you will immediately be removed from the webinar. Lastly, we ask you to bear with us if there are any unexpected technological difficulties or distractions. So first up, we have Dr. Karen Fong. Dr. Fong leads a food microbiomics research program that is focused on investigating pathogen mitigation, including Salmonella verotoxin producing E. coli. Her group primarily uses omics technology to characterize novel approaches to ensure the safety and security of the Canadian food supply. Additional research interests include food and environmental metagenomics, antimicrobial resistance, and microbiology. Um, of fresh produce commodities. Karen was born and raised in Okanagan region of BC. She holds a PhD in food science from the University of British Columbia and was a recipient of the Canadian Dairy Commission and NSERC Alexander Graham Bell Canada Doctoral Scholarship. So welcome, Karen.
Hi, good morning, everyone. I'm going to share my screen here. Okay, so just verifying that you can see my screen. Okay, perfect, thank you. All right, so, just move things around here. Not looking at myself. Okay, excellent. Good morning, everyone. My name is Karen Fong, and I am a food safety research scientist / microbiologist with Agriculture and Agri-Food Canada. Before I get started today, I would like to acknowledge that I am calling in from the interior of BC, from Kelowna, and I am on the traditional ancestral and ceded territory of the Siouxsie people of the Okanagan Nation. The title of my talk today is Going Viral, a novel farm-to-fork strategy for fresh produce. And I'll be touching upon some select studies that I conducted throughout the course of my PhD, as well as into my postdoctoral training. To begin my talk today, I would like to present you with a snapshot of foodborne disease in Canada. So it's estimated that roughly 4 million Canadians are affected every year by some form of foodborne illness. And oftentimes, this illness will manifest in a variety of different gastrointestinal symptoms. However, there are some illnesses that can result in more severe symptoms, such as muscle paralysis, reactive arthritis, neurological damage, and even death. The majority of these illnesses are caused by various microorganisms, including bacteria, viruses, and parasites. And some of you may be familiar with some names that have been probably tossed around in media, such as norovirus, campylobacter, salmonella, E. coli, and listeria. And some of you may also be familiar with their source attribution, so the types of foods that these microorganisms are associated with. For instance, salmonella in chicken, E. coli in beef products, listeria in ready-to-eat foods. So there's a variety of different foods that may be implicated. And although they can, increase the disease burden within Canada, a lot of times they result in very costly recalls of the food product itself as well, costing the Canadian economy billions of dollars every year in loss. So as I was doing some reading in preparation for this seminar, I came across this interesting article, 3 things that food safety experts never eat. So I decided to read about it a little bit further. And there were some obvious ones, such as raw unpasteurized milk. Raw unpasteurized milk is strongly associated with E. coli. And even in the Okanagan, there have been outbreaks of E. coli in raw milk cheeses. Another food that was mentioned was raw shellfish, such as raw oysters, which are a potential reservoir for Vibrio perihemolyticus. And very interestingly, one of the foods that this particular food safety expert mentioned he would never touch is sprouts. And this is kind of interesting. And today I want to focus more on sprouts because, they're not commonly associated with foodborne illness. Not a lot of people would typically associate this as a vehicle for salmonella or E. coli or listeria. However, sprouts are actually increasing in popularity, particularly in Western countries. And oftentimes, they're kind of considered more trendy. However, they are considered also a higher risk food commodity. And today I will touch upon why that is. So as I mentioned, sprouts are considered a type of health food. They're high in vitamins, they're high in minerals and other nutrients. They're high in healthy gut probiotics. However, they are characterized as a higher risk food commodity. And for those of you who regularly consume sprouts, you could probably see why, because, the typical shelf life of sprouts is not very long. You buy sprouts, you put them in the fridge, you might forget about them. And then a week later, they're sort of soggy and kind of disgusting looking, and you wouldn't really want to eat it. And I'll just talk a little bit about, the sprouting process. So how to actually obtain a sprout. So this is, this diagram here is sort of what happens is you start off with a seed, and then over time throughout the sprouting process, the seed eventually will sprout something, which is what we eat. However, this process is actually dependent on a variety of different factors, such as adequate water, high humidity, and even warm temperatures. And coincidentally, these conditions are actually what we use in the lab to get bacteria and other pathogens and other microorganisms to grow. So it's essentially acting like a giant incubator. And in these conditions, any pathogens or any disease-causing agents that are present on the seed can actually start to grow as well. So what's potentially lurking in our sprouts? Well, one bacterium that has been causing hundreds of outbreaks is Salmonella. And Salmonella can cause a variety of different symptoms in humans: gastrointestinal discomfort, cramping, pain. Serious infections can lead to reactive arthritis. They've been linked to deaths. And producers are aware of this. So producers are aware that Salmonella may be present in the seeds themselves, and they can use a variety of different chemical treatments to mitigate this risk. And one very common treatment is actually to use chlorine bleach. However, it's not exactly clean label. So you won't be able to necessarily infer that it was organic. And oftentimes, especially considering the mindset of a lot of people eating sprouts, you wouldn't want to eat sprouts that have been treated with bleach. So one alternative to using chemicals in not only sprouts, but other food commodities as well, is to harness the power of bacterial phage, or phages for short. So this is a picture of a typical phage, and it kind of looks like this weird sort of spaceship thing. But briefly, phages are viruses of bacteria, but they don't infect human, animal, or plant tissues. And phages are actually ubiquitous, meaning they're everywhere in the environment. They're found on our skin, they're found in our gut, they're found on food already, they're found in soil, and phages outnumber bacteria 10 to 1. So for every one bacterial cell, there's at least 10 phages that are ready to infect it. And if you just think about, the ubiquity of phages in the environment, it's actually thought that every 2 days or every 48 hours, the global bacterial population is reduced by 50% because of bacterial phages. Just talking quickly about the different structural components of these phages. At the very top here, you have what's known as a capsid, which is essentially just a protein head that encapsulates the nucleic acid. In tailed phages, often it's DNA. And then at the very bottom here, we see tail fibers here. And these tail fibers are the recognition component of the phage. So this is what actually recognizes bacterial proteins on the surface and uses them to attach. I really like this picture as well because this is an electron micrograph of a bacterial cell that has basically been killed off because of phage infection. So essentially lysed the bacteria from the inside out. And as you can imagine, phages are used for this purpose in food production. So phages can be used for biocontrol. The first being is that they're sustainable, they're found everywhere in the environment, they're readily sourced from the environment. Phages are very pathogen specific. So, if you find a Salmonella phage, it'll only infect Salmonella without infecting other bacteria, without infecting human, plant, or animal tissues. And very importantly, they don't actually alter the organoleptic properties of the food. which means that they don't change the taste, the smell, the aroma, or the appearance of food. So they're very good candidates in terms of food production. So essentially, during my PhD, this is what I wanted to find, was to find a phage or several phages that would be good candidates for food biocontrol. So I went on a phage finding mission where I found myself in very, let's say, interesting environments throughout the course of my PhD. So these environments included soil, irrigation water, poultry farms, cattle farms, fish farms, and ultimately what I was trying to do was I was trying to find some superphages. And so I found this particular phage known as phage SI1. And this phage was taken forward into several other studies because it showed a very good Salmonella host range, which means that it was able to infect several different types or strains of Salmonella. And it was also temperature stable as well as pH stable, which would make them good for food production purposes. So one of the first studies that we did was we wanted to see how well it actually infected Salmonella in broth culture. So when I say in broth culture, essentially what I mean is, in a lab-oriented setting where conditions are perfect for growing bacteria and for growing phage. So this figure here essentially shows the OD600, which is on the y-axis, and OD is simply a relative measurement of the cell density in whatever medium you're trying to measure it in. And then it's given as a function of time in hours. And then this line here at the very top with the blue arrow where it says Salmonella is essentially the Salmonella cells that are growing uninhibited. So this is like a perfect scenario. So you see rapid growth of the Salmonella in the broth culture. However, when we add phage SI1, you can see that it actually suppresses Salmonella growth very, very rapidly over a time period of 35 hours. So already we're seeing, very good suppression and very promising results. However, we wanted to transition what was shown in broth culture into more of a, I wouldn't necessarily say field setting, but more of a real world situation. And so we took it a step further and looked at and analyzed how well this particular phage did in sprouts. So we started off with alfalfa seeds and we basically artificially contaminated it with Salmonella. And this is how it would typically be done in real world settings, is you would have seed that was contaminated with a pathogen. Next, we added our phage SI1 to the seeds. And then we observed how well the Salmonella behaved with phage infection over 6 days of sprouting, which is what's typically represented in real world settings. So here again, we see that phage SI1 actually does suppress Salmonella in sprouts to a certain extent. So this graph here looks at essentially Salmonella suppression over a period of 6 days, which is the sprouting period. And I just want to sort of draw your attention to day 1, because here we see the greatest reduction in the Salmonella with the phage treated seeds. We actually see a 2.5 log CFU per gram reduction, which correlates to around a 99% reduction. So, this suppression is most pronounced early on. However, from days 2 to 6, we actually don't see that much of a difference. And this does warrant further investigation, but we hypothesize that bacteria may be gaining some sort of resistance to the phage treatment because we know that the adaptive response can kick in. So there are other strategies that can be used to actually circumvent this phenomenon. We could go forth with repeated application, or adding different or diverse phages. And however, due to time constraints, I won't be able to discuss those in full. So I'll leave it here and perhaps we can touch upon it in the Q&A period. So lastly, I'll leave you with some conclusions and some take-home messages. So it's been shown that sprouts and other produce commodities, such as other fruits and vegetables, can be readily contaminated by human pathogens. However, phages can be used as a mitigation tool, a chemical-free alternative, because they have several different desirable properties, such as sustainability, host specificity. They're not, a danger to human, plant, or animal tissues. And it's actually interesting because this particular phage that was talked about in this study, phage SI1, was actually incorporated into a produce treatment that is currently undergoing larger field trials in the US. And as with any area of new technology, research is currently ongoing, especially to acquire new knowledge about effective phages and to better characterize bacteria and phage interactions, and to collectively gain a better understanding overall of how phages can be most effectively used in the food industry. So lastly, I would just like to acknowledge the Wong Lab of Molecular Food Safety, which is where I did my PhD, as well as my postdoctoral training, especially my supervisor, Dr. Sian Wong, as well as my funding sources, UBC, the Canadian Dairy Commission, as well as NSERC. Thanks very much, and you may reach me at this email provided here if you have any further questions, and looking forward to chatting more in the Q&A.
Thank you so much for that wonderful presentation, Dr. Fong. So next up, so please, people, if you have more questions, just put it on the question and answer tool in Zoom. Next up, we have Dr. G.T. Cornelis. J.T. is a pedologist and biogeochemist, assistant professor in soil science at the Faculty of Land and Food Systems, University of British Columbia. J.T. comes from the University of Liège in Belgium, where he was appointed as an assistant professor in soil science. He received a PhD from the Earth and Life Institute and University of Louvain in Belgium too in 2010. His research investigates soil processes controlling biogeochemical cycles of nutrients and how soil-plant feedback interactions respond to environmental changes. His process-based approach focuses on the soil system, which is by nature multi-scale and interdisciplinary. JT takes as an advantage the complexity and diversity of soil processes to teach students the notion of critical thinking and complex thought. Daddy of two little boys, outdoor enthusiast, big fan of surrealism, he loves to brainstorm new ideas. So welcome, JT.
Thanks, Aida. So let me share my screen. Okay, can you see my screen? Okay, perfect. Thanks, Aida, for the introduction. I'm so delighted to be here with you. So what I'm trying to do today is to exemplify why we should maybe harness a bit more the complexity of soil-plant interactions to build resilience in agroecosystems. So first of all, Soil Westry Lab at UBC acknowledges that we are gathered on the traditional, ancestral, and unceded territory of the Musqueam people. So almost all soil-related environmental challenges are a question of spatial and time scale. And I like to remind here that if we are bringing back the Earth's age to a one-year scale, the Anthropocene started in 1785, has completely reshaped Earth's planet in less than 2 minutes. So yes, needless to say that we have few seconds to act. And so you can see in this map that Earth's planet then emerged lands in 1700, were almost entire semi-natural and wild. And in only 300 years, almost all the biosphere is man-made affected, with less than 45 semi-natural and wild territories left. So what we can see on this very specific graph, on the y-axis, you have the yield level, and on the x-axis, the land use intensity. So obviously, if you're increasing the land use intensity, for example, by getting more mineral fertilizer, you will increase the yield of your system. And so this is why we are still observing 25% lower yields in organic farming compared to conventional farming. But we have hope here because using some soil ecological engineering, we can maybe replace this use of external inputs. And even with some very specific soil ecological engineering processes, we can even enhance the yield in the agroecosystems. So, and with regard to this soil ecological engineering practices, we know that there are some at the macro scale. I'm talking here about cover-crop association, but there is also some micro scale engineering process that we can put further in terms of more efficient, resilient, and sustainable agroecosystem. And for this, I think that's pedology and pedologist is really, really useful. I like to point how pedology background is of utmost importance, especially with respect to the urgency we are facing because of human-induced global changes. So no, pedology is not a vintage car. I think pedology is a vintage wine. And so why? Because the pedologist can give maybe to the people a better view of the environmental factors that are responsible for soil formation. And so we better know why we are observing this kind of soil in this very specific place and not in another one. So I mean, we can understand soil constituents, soil properties, and relationship with soil processes. And the challenge of the next generation of pedologists, I think so, will be to develop strong skills in pedology, soil genesis, together with growing curiosity to other disciplines to acquire new skills in applied geochemistry, microbiology, as presented by Karen just before, plant physiology and ecology. And doing this, maybe we can move forward, the field forward when combining pedology background with the other disciplines. So to achieve this goal, it's urgently needed, I think, to think out of the box because soil and agroecosystems deserve really better than the still too common practice of evaluating plant response to mineral fertilizer without relating to that to the soil properties and soil root interaction. So that's maybe the black box vision, and we have to move from this vision to another vision, more integrated in terms of disciplines, but also in terms of scales. So I mean here, if we are adding something on soils like mineral fertilizer, we have to go further in terms of evaluating the plant response instead of only evaluating the yield without knowing anything in terms of how soil processes can influence or can be influenced by these interactions. So yeah, for sure, we have to shed a light on the soil complexity by interdisciplinary and multiple scales approaches. And one soil hotspot that is maybe of huge interest is the rhizosphere. And rhizosphere is the soil region directly affected by root secretions and associated soil microorganisms. So it's hard to imagine how much this kind of hotspot region is a super innovative factory which naturally succeeded to develop strategies for plant bioprotection, for sure, but also strategies to acquire the nutrients essential for their growth. So you can see on the picture on the right, so the rhizosphere on the left is this very hotspot zone associated between soil and roots. And so on the right, you can see that the more you have soil and root interactions, the better will be your phosphorus, zinc, nitrogen, manganese, iron, water acquisition in the system. So the availability of water and nutrients in soil varies a lot in time and space, and is often predictable. So this is why soil exploration is therefore a primary challenge in plant biology to get root plasticity that can optimize soil resource capture in patchy soils. So especially under increasing biotic and abiotic stresses for staple crops. And one point that I want to stress here is in terms of root traits with regard to soil exploration is the exudates release, so the secretion of organic molecules by root systems. So the challenge is that all of this kind of plant-induced mechanisms like root secretion are intensively studied in highly controlled lab conditions. So what I mean here is that it's very simplified condition. And this is normal because the scientist has to understand the system. So we need to isolate the confounding factors. But we know also that soil are very diverse. And so the different color you can see in this soil profiles reflects the presence of different mineral and organic constituents that in turn control soil properties such as texture, structure, pH, nutrient content, and availability. So for succeeding in plant breeding, which is very yield performance targeted, because plant has to cope with biotic and abiotic stresses for sure. But I think it's urgently needed to have new breeding strategies, which should be more soil ecosystem driven. What I mean here is that a plant genotype won't respond the same way depending on the type of soil constraints. For example, in terms of constraints, deficient soils in certain macro or micronutrients, or pH challenging soils because too acidic or alkaline, or structure texture challenging soils because too sandy or clayey. So I mean here the plant response really differed between these different type of soils. So the current plant breeding strategy is still certainly too much rooted in black box soil vision. So getting the right production targeted genotype, which we will feed with food. I mean here nitrogen, the right dose of nitrogen, phosphorus, and potassium, together with kind of medicine for digestion. So I'm talking here of liming the soil to increase the pH to get the very right bioavailability of nutrients. So this is the way we are dealing now with soils, more like a physical support. But we have moved from this black box vision towards embracing the full complexity of soil properties when we are dealing with genotype and plant breeding in the future. So I just want to take the opportunity of the next 4 or 5 minutes to exemplify why we have to maybe better interconnect soil and plant systems in agroecosystem. Look at that. So I don't want to go too deeper into the details here, but what you have to keep in mind is that when you are going in the soil profile from the left to the right, you are increasing the soil degradation. So this is a soil chronosequence we studied with Félix de Tomber, a PhD student from Belgium in Western Australia. And these soils are very young on the left, so with a high content of nutrients, and very old, 2 million years old on the right, so very leached. All the nutrients content, calcium, magnesium, are leached out of the soil profile. So this kind of soil are highly degraded and depleted in nutrients. And what we can see as we focus our study on silicon specifically here is that the availability of silicon in soil obviously decrease with the degradation of the soil. Why? Because you are just losing silicon in your system, and the only remaining constituent in soil is the quartz. So it's a very typical mineral that is very, very tricky to dissolve, very resistant mineral. But we have been really, really surprised and also excited to observe that when we are quantifying nutrients in plants above the soil, in the above ground biomass, obviously we can see that all the major nutrients like calcium, potassium, magnesium, and phosphorus are decreasing with increasing soil degradation. Pretty normal and logical because all these elements are leached out of the soil profile. But what we have seen in purple here is that silicon content is still increasing in plants with soil degradation. So the hypothesis we put together here, beside the role of the biocycling of silicon in soil plant system, is that plants can cope to develop some very specific strategies from the roots to acquire silicon. And why? Because silicon, if silicon is accumulated in leaves, this is a way to protect the plants against abiotic and biotic stresses. And what we observed by doing a very specific lab experiment in controlled conditions is that, yeah, this kind of plants that can release some carboxylates, some very specific organic molecules in roots, can cope to dissolve silicon from the minerals and to accumulate this silicon in leaves. So this is pretty amazing to see that this kind of soil-plant feedback interactions and the way the plants are able to develop very specific root system, as you can see on the picture on the left, in very degraded soils, is really, really something that we have to invest, if I can say, in terms of agroecosystems, because this kind of very specific root systems and nutrient acquisition strategies can help plants to acquire some very specific nutrients, as we have shown for silicon. And this is certainly a challenge we have in front of us. Why? Because we really need to now to translate this kind of processes we observed in natural ecosystem in agroecosystems. And so this is maybe, yeah, simple to say, but to traduce it in the lab and in agroecosystem, it's certainly difficult to do. But this is a challenge we have to do and we have to respond for, because on top of that, they are very, we can be very excited because what we observed for silicon is certainly true for nitrogen and phosphorus and potassium, as already demonstrated, but also for micronutrients. I'm talking about molybdenum, zinc, iron, and manganese. So definitely this kind of root-soil interactions and strategies for acquiring nutrients can be a game changer in agroecosystems. And so to conclude, the urgency now, and this is pretty urgent because of the environmental and climatic challenge we are facing now, we have to translate the mechanisms that we can observe at a very micro scale in soil. So I mean here at the interaction between roots and soil particles. We have to translate it, what we will observe in Weizen boxes on the left picture of the slide. We will collect soil solution and we can measure carboxylate organic molecules in the soil root solution. To understand this kind of interactions, we have to translate it in soil profile and then finally to field settings. So this is our duty as pedologist and biogeochemist. And I want to thank you now because that's all. So you have my details here and also the link to the website of the Soil Westry Lab at UBC. Thanks.
Thank you so much for that wonderful presentation, JT. Okay, So now it's time for us to move to the question and answer portion of the webinar. I'll be asking our panelists some curated questions as well as any live audience questions we receive. A reminder to navigate to the Zoom control panel and select question and answers to input your questions as well as about other questions. So the first question here is, what's the accessibility for small scale food producers to access this phage technology? Karen.
Hi, yes, thank you. Thank you so much for your question. So so phage technology is still very much in its infancy, not only in Canada, but worldwide. There are some commercialized phage products available on the market, depending on the food that's being produced, that has been validated for use on different foods. For instance, I think it's a product called Salmo Fresh, and it's a phage cocktail, which basically means like a mixture of different phages that have been validated for use, I believe on poultry products as well as some produce items. So it does depend on, again, the type of food that's being produced here. In terms of host range, and I know I didn't really elaborate too much on that in my talk due to time, but in terms of host range, Salmonella is it's a very diverse pathogen. And so oftentimes you'll find different types of Salmonella being associated with different types of food products and in different plants. So my advice for this person would actually be to, figure out specifically what types of Salmonella are contaminating your facility, or what types of Salmonella is of greatest concern to you, and then actually reaching out to government, actually, because there's a lot of phage biologists in government, myself included. And there are, I believe, some technology grants available through the government that can be accessible for smaller scale producers.
Thank you for that answer. So the second question that we have here is thinking of the microbiology of the soil and the microbiology of food safety, how do we bridge this gap to start healthy, contaminant-free growing in the field?
Maybe I'll go first, if that's okay with JT. I think this is a question for both of us.
Yeah, sure, go ahead.
Okay, this is a very, very good question. And when I saw it, I actually had to think a little bit about it. So one of the tools that microbiologists kind of have up their sleeve is metagenomics. And metagenomics is a field of research where we essentially analyze the entirety of the microbiome of a given environment. So using metagenomics, we can generate knowledge of various, microbial interactions that are going on in soil, with the hopes of enhancing soil health, plant health, as well as food safety and food security. And just kind of thinking about this a little bit further, it's been shown that differences in soil, microbial differences in soil, may be a predictor of soil health and also risk for pathogen contamination even, because it's been shown that certain pathogens such as E. coli and Shigella are more likely to colonize different types of soil based on the abundance and diversity of the soil microbes already present.
Well, sure. And I just want to add something here. We have also here a very nice opportunity to connect also soil microbiology with soil chemistry and soil physics and plant science, because we know that in the rhizosphere, we have very specific interactions between microbiomes and root system in terms of plant bioprotection for sure. But we know also that some specific microorganisms release also, like the roots, some exudates that can play a key role in nutrient acquisition. So yeah, for sure, we have an avenue here to connect the different disciplines.
Thank you so much for that. We have another question, and is increasing silicon necessarily evidence of a plant strategy? Why have strategies for getting silicon and not micronutrients that are arguable more badly needed?
That's a wonderful question. We got this quite a similar question during the review process of our paper. So yeah, that's wonderful. And so this is a question of the cost for the plants. So it's less costly for plants to acquire and accumulate silicon, because that's like accumulating biogenic silica, like a stone in the leaves. And this phytoliths accumulation in the leaves can help to protect plants against biotic and abiotic stresses. So getting silicon in plants is consume less energy for plants than accumulating phosphorus or other elements. So yeah, in this very specific environment in Australia with highly degraded soil, this is what we observed.
Great. Thank you for that. And then the last question, or well, next question is, what assumptions are you making about water availability, like drought versus flood, in your soil experiments, along with evapotranspiration rates in climate change conditions, for example?
Yeah, so that's a very interesting one. Thanks. So interesting one, because we cannot separate water dynamics in soil plant system with nutrients dynamics. And so together, we know that if we are improving the nutrient use efficiency in the soil plant system, for sure, your efficiency of using water in soils will be improved also. And so if we are succeeding to get better root system, root traits in soils, it means that maybe the plant can cope better drought events in the next few years. So this is the way we really want to go through with the people here in the lab, because we know that better root systems and root traits can improve the nutrient acquisition, but also store organic matter in soils, because we have the above ground of the biomass, but also the below ground biomass is a very important pool in terms of organic carbon. And on top of that, we know that if we are improving the stock of organic matter in soil, we will improve the water use efficiency and water storage in soils.
Great, interesting. So extension is such a key part of getting this incredible research from academic institutions and government labs to the people that actually need the information, like the farmers. How do you do extension, and where do you see further opportunities? This is a really, really interesting question that I think that all academics and researchers get all the time. So please go ahead.
Karen.
Oh, sure. Thank you.
Sure.
Thank you so much for this question. I agree. It is very, very important. So myself personally, I work out of the Summerland Research and Development Center, and there we actually have a biologist who is a knowledge transfer specialist. So I work very closely with him in getting the results from my work across to various sectors, various industry groups. Here in the Okanagan, we're primarily working a lot with cherry farmers and with stone fruit farmers, smaller scale producers. My colleagues at the Agassiz Research and Development Center work more with dairy farms and with poultry farmers. So, all the research branches within AAFC are sort of uniquely positioned and sort of have like a different sector with which they directly interact with. Lastly, I'll just quickly mention, we do also work very closely with these different sectors and talk directly to them to identify their needs, which further informs our research direction and research avenues.
Yeah, wonderful, Karen. And so on my end, as soil scientist, that's really extension. Yeah, I agree, is a key part of the game. And so, as I always said to the researcher in the lab, we don't have to lose too much time in the lab. We really need to invest also time on the field. And so, yeah, just to make a long story short, I can give you just one example of a project we have in Burkina Faso, and what we did in this project to be sure that the technology that we want to use to improve soil functioning, this technology was the biochar, a mixing of biochar with manure, is that we are doing some living labs, field trials with the farmer. So we did very field experiments in very controlled conditions to test the different treatments. And then when we get very good data about this kind of treatments, we expand this directly with the farmers, doing this kind of living lab trials directly on the field. And so after 3 years of trials, we succeed to get very interesting data in terms of crop yields, but also in terms of soil chemistry and soil biology parameters. So yeah, that's an urgency to proceed this way, I think so.
Great. The next question is, with the COVID-19 pandemic, microbes and the microscopy world have become to the forefront of our thoughts like never before, especially for Dr. Fung, but also generally, how do you see this new microscopic approach affecting how we handle our food supplies? Right? I think that this is the time that people are thinking about
Yeah, definitely. And that's a very, very good question. So I remember at the very beginning of the pandemic, there were a lot of food producers and food manufacturers becoming very concerned with how the SARS-CoV-2 virus, which causes COVID-19, can contaminate their food. So not only food, but also food contact surfaces, such as cutting boards, knives, food packaging. And so there was a lot of research that happened right at the very beginning to identify, was SARS-CoV-2, is it a foodborne pathogen? And so one of the criteria for being a foodborne pathogen is that it actually has to survive in food. So at the very initial beginning of the pandemic, a ton of research was funneled into this. And it was, it's now been conclusive that SARS-CoV-2 is not a foodborne pathogen because it cannot survive in food products. What's really interesting about this is that, given that I work with viruses and SARS-CoV-2 is also a virus, there is naturally some sort of interplay and some sort of overlap between these two ideas. And so I recently just finished a project where we actually used a bacterial phage, which is known as phage MS2, as a surrogate for studying SARS-CoV-2. And essentially what a surrogate is in microbiology is it's a pathogen it's a microorganism that you use that is less dangerous than actually handling, for instance, in this case, SARS-CoV-2, so the actual COVID, virus. However, it also, you're kind of limited with what you can use it for. So in this case, I was using this particular phage MS2 to study surface inactivation. So to study how resistant it was to commercial sanitizers that are used in the food industry, such as quats or alcohol sprays or bleach. So I find this question very timely and also very interesting because it's certainly enabled us to sort of think of food safety in a different aspect, instead of like the traditional ways that we would typically approach a food safety problem.
Great. Thank you so much for that. Our very last question, and we apologize in advance because we are not able to answer all of your questions, but we can reach our panelists today asking for deeper explanations. But the last question is, sometimes the solutions we found in the lab are unreasonable, expensive. Testing your soil for microbes or genetic testing to identify pathogens, breakdowns, the barriers to these technologies and create a more equitable distribution of resources. What are your thoughts about it?
Yeah, that's really an exciting one. Yeah, thanks. I would say that all the solutions are already existing in the nature. So we just need to translate what nature can learn us and directly for the agroecosystem. So yeah, I'm just saying here that personally, I'm a firm advocate of nature-based solution for climatic questions, climate mitigation, and agriculture adaptation to climate changes. So what I mean here is that obviously in the lab, we will do some soil chemistry analysis, also some genomic analysis with the colleagues here working in plant genomics. So that's our duty to carry out this kind of experiment in very controlled conditions to understand the mechanisms. But when we come up with very interesting solution, for example, a type of plant or genotype that can release a huge amount of organic molecules from roots to acquire nutrients and to store organic carbon in cells, the translation in the field is pretty easy because that's a nature-based solution. So I think that's maybe the way we have to go forward. And so, yeah.
Yeah, I don't have a good answer for this question, I'm afraid. Yes, research is very expensive. Research is very time consuming and very costly. I will say though, some of the, what we consider basic services now, for instance, sequencing. So, identifying the entire DNA sequence of your organism of interest, for instance, has gone down a lot in price. So it is quite cheap to sequence a microorganism or sequence, a certain environmental sample now. But really, this is sort of one of the things that is definitely a barrier in our fields. So, unfortunately, I don't have a great question or a great answer for you. Yeah.
But we have to start from somewhere, right? So I think that we are in the right place, and I think it's fascinating how in such a short time vaccines are available, and this is because of research, right? Otherwise, we wouldn't be here right now with classes in person again in campus. So yeah, I think that any kind of research is gonna give us a step forward or goals in a short future, hopefully. So thank you so much, everybody. Thank you, JT and Karen, for these wonderful presentations and the answers provided today. And as I mentioned before, there are so many unanswered questions, and we thank you all for sharing your time with us. Keep your eye out for a follow-up email, which will address some of these questions and share resources what we have learned today. So thank you again, Karen and JT. Behind the scenes, we would like to thank Melanie Kuxdorf, Mairead Norton, and Julia Taylor. Thank you so much for putting all this together for today. Big clap for you. And please join us next week for our final episode of titled Regrowing Agriculture. This is going to happen on September 16th at 11 a.m. again. And let us know what are your thoughts about these webinars, and you'll be directed a short survey about this session after the webinar. So we really appreciate your feedback. Also, feel free to visit us at ubcfarm.ubc.ca. Thank you so much for joining us today, and we look forward to continue the wonderful discussion.
Thank you. Bye-bye. Take care.
Thanks.
Thank you, everybody. Bye-bye.